Translation. You have the message. Now you need to decode it. That's translation. Translation is how cells convert the message into proteins. It happens at the ribosome. Here is the process. The ribosome is made of two subunits, large and small. The mRNA threads between them like a tape through a cassette player. The mRNA is read in groups of 3 bases called codons. Each codon codes for one specific amino acid. Transfer RNA or tRNA brings amino acids to the ribosome. Each tRNA has 2 important parts. One end carries a specific amino acid. The other end has an anticodon that matches a specific codon on the mRNA. Translation begins at the start codon AUG. This always codes for the amino acid methionine. The ribosome holds the mRNA in place. A tRNA with the matching anticodon binds to the codon. It delivers its amino acid. The ribosome has 3 critical sites: the exit site, peptidal site, and amino acyl site. The amino acyl A site accepts incoming tRNAs. The peptidal P site holds the growing protein chain. The exit E site is where empty tRNAs leave the ribosome. Each one of these plays a major part in translation. The ribosome moves forward by one codon. Another tRNA arrives with the next amino acid. The ribosome forms a peptide bond between the 2 amino acids. The first tRNA is released. The chain grows. This continues codon after codon, amino acid after amino acid. The protein chain extends. In bacteria, the ribosome moves along the mRNA at about 15 to 20 amino acids per second. When the ribosome reaches a stop codon, UAA, UAG, or UGA, translation ends. The protein is released. The ribosome subunits separate. The genetic code is nearly universal. The same codons code for the same amino acids in bacteria, plants, and humans. This is one of the most profound pieces of evidence for common ancestry. Translation is the final step of gene expression. The RNA message is decoded, a protein is built, the instructions stored in DNA become biological function. Translation converts mRNA to protein. It occurs at the ribosomes. They are composed of large and small subunits. Codons are 3 base groups on the mRNA. tRNA acts as the amino acid delivery trucks, matching anticodons to codons. AUG is the start codon, which always codes for methionine. The stop codon can be UAA, UAG, or UGA. Translation runs at a speed of approximately 15 to 20 amino acids per second in prokaryotes. It might be slower in eukaryotes. The genetic code is nearly universal, pointing to a common ancestry.